All right. I want to thank you for inviting me here to talk. My session is called Failure to Launch. And I'm going to describe a little bit of what I call the history of biotech in one slide. And that's this slide. It had its origins at the first talk here in 2024. And so what I'm going to do is kind of walk through this, and how I've evolved it over time is to use the idea of what happened with, there's a fun thing, is what happened if you look at from 1900 to 1960, we went from Kitty Hawk to the moon. So I'm using that as an example of what kind of a technology evolution could happen with an initiation going to a remarkable culmination. So right after we landed on the moon in the early 70s were these infamous disclosures of work by Berg, Cohen, and Boyer that really kicked off the technological framework for modern biotechnology. So what I've done then is kind of said, well, let's go from 1970, let's use that as the kickoff. Where would we be in the same time frame at the end of 2030 with biotechnology? Are we moving at a pace where we could say, oh, we've launched? Or, as I've argued, in some areas where we've failed to launch. So I've got these organized in some bars. We've got Nobel Prizes, policy and regulation, legal, biomedical, non-biomedical FDA, as we've talked about it, and then environmental and bioindustrial. So I'll go through it relatively quickly. These are just some of the keynote Nobel Prizes. I need to probably go and add computational protein design with the Baker thing, but you can see pivotal, fundamental science has moved along as this field has developed. So I think we can go, yep, we're checked. We're doing something there. As we've talked about, we've hit Asilomar. We've hit the coordinated framework. All of these, I put them in a kind of policy regulation framework simply because they kicked off after the Asilomar moratorium and moved forward. MCAN may not be as familiar to some of you, but then I know a couple of organizations here that know what the MCAN is. I did modify this to indicate one of the things that did happen. So after the secure rule finally got established, it was vacated by a court ruling. And so I'm not trying to be comprehensive. I'm just trying to show some things that really looked at the kickoff. Fun. We have also mentioned Chakrabarty. And the SCOTUS ruling on that really kicked off a legal framing that allowed a lot of this work to move forward. What is interesting, again, this is General Electric. What in the world is General Electric doing with what we call modern biotechnology? If you use an alcohol-inducible gene in a yeast, think Phillips Petroleum. What was going on back at this point in time where a lot of companies that we don't see as part of biotech industry were heavily involved in it? And this, of course, is the development of restriction into nucleases, which was critical for this to move forward. So now let's look at the biomedical framework. We've got Genentech and insulin going to Eli Lilly and their recombinant insulin. Genentech at the same time was moving in human growth hormone. That got approved through the FDA. Genentech also did bovine somatotrophin. We can call that a somatotrophin, but that's RBST. So now we start seeing this framework where these are widely accepted, don't have public negative perception, but RBST that was moving along at the same time actually ended up with significant negative perception. One of the things that's interesting here, FDA over this time frame approved 100 monoclonal antibodies. And so when you think about something where you are able to scale, is this system able to be replicated? Can you come in and do new things? Even if you remember the Ebola crisis in Africa in the late 2010s, the monoclonal antibody that was used for that came out of a transgenic tobacco. So even something unlike the normal CHO or other cell frameworks, EPA or FDA was able to handle that. So 100 antibodies in that time If you kind of jump down here, that's the time for the salmon growth hormone that we were talking about on earlier slides. Now, one thing that I like to point out, because we just don't think about it, that's 80 or 90 amino acids. That's a couple hundred, 150 or so. Chymosin from genes was a little bit larger. All of these had in common being small enough to be able to be synthesized by the gene synthesis technology of that day. So cloning was important, and we did find the genes by cloning, but these almost invariably were moved in through by gene synthesis at the time. And that was important in part of their approval and in the evolution, say, of the chymosin framework for genes, because you could make a vegan version. You could start changing the variations of this that were important for public perception when they even knew it. And so then this timeline as we look along here. So RBST was actually finally approved. These were companies that were involved at the time: Monsanto, American Cyanamid, Eli Lilly, Upjohn. Everybody was trying to play in the game. It was the largest investment in biotechnology outside of medicine at that time. Public perception, real issue. This, as we talked about before, finally came. Now, FDA also did some things. You've got the glowfish that came out, and then later with EPA, the firefly petunia finally came out. So some novelty things that were moving directly to consumers were able to make it through processes. But I think what you can see along, and here's GLP-1 type things, all right? That's the whole time frame from going from a phase 2 clinical trial to when something was able to be marketed. Has FDA and have the actors in the field of biomedical biotechnology figured it out? I think you can say yes. I think you can say they have launched. And again, in the non-medical applications where FDA is a large player, even though there's some things with long time frames, you can still figure out and meander through it if you have the stamina, the staying power, the financial backing to figure that out. Not saying that's good, just saying that that's a reality of the situation. So now let's jump down here. And this is the place that I like to talk about. I was working in this area. We actually got the first dead recombinant organism as a biopesticide through the EPA in this time frame. So this was all very personal, very intimate to me. This was framed out by looking at the ICE- framework. And I've got a little picture here. We'll get into it later. But if you don't know ICE-, you've got to go learn ICE-, because it set the framing for how things moved here. It got through the EPA, then got into court with a lawsuit, then got into field trials. The field trials actually happened, but there was no ICE- product on the market for a while. We can talk about that in a minute. But all along here, the bioremediation, biopesticides, biosensors, all were being developed in this time frame, and they kind of came to a halt, I'll call it, in the mid-90s. Part of this moved, in terms of the biopesticides, moved from microbes or insects as pesticide delivery entities to the in-planta, so the GMO crops that had the insecticidal proteins in them. And these moved forward in the mid-90s, also the recombinant technologies for herbicide tolerance. That moved. As Richard had already mentioned, you have this framework coming out of the MCAN where the terms intergeneric and intrageneric became important. And as we mentioned, TSCA and FIFRA became important. And so we won't go into all this. There are people more qualified to me to get into the legal aspects of this. But those were all important things that were happening in this time frame. Of late, we've had renewed interest in biofuels, biochemicals, biomaterials, largely in contained processes, so inside a bioreactor somewhere. And then biofertilizers, that would be code name for pivot and a lot of other things that have moved there. So these are all moving. So what I'm going to do now is go to something. I want to try to put together some historically contextual documents just to go digging. This was in Science. What's nice about it is this is 1987, and this is a one-pager describing all of the things that were in the environment or trying to move into the environment as a result of product development at that time. So these largely all had a product development framework. We mentioned earlier, someone mentioned the idea of an organism that had a special relationship with the plant. Well, that's what got the rhizobium thing through the reviews, its tight association with the plant. People were trying to get lac containing organisms all into the field. These challenged the review process, and several of them were denied. This recombinant insect control was the thing. It was a single gene thing. Everybody knew the genes, and it seemed like the perfect scenario to be able to use recombinant DNA technology. And again, some things got into the field and some things never made it into field trials. Bioremediation, that HK44 that was on one of Emma's slides, it was the longest running field trial release, but the majority of these that were envisioned by the Chakrabarty patent just while they were on the doorstep in '87 did not make it out. And again, we had other frameworks, recombinant baculovirus and fungi for disease control. So there was a lot of stuff going on in advanced field development at that point in time. This is from a series of publications. This was the third series from the OTA, the Office of Technology Assessment. That's the logo. This was a series produced in the 80s and early 90s. It's wonderful. This is a link to higher quality images, but the whole series is hosted on a Princeton website. Otherwise, it's kind of like lost in a library somewhere. But what you can see from this, these were organisms and concepts that were in play at this point in time, trying to get into field release or further development. And so you can see all the kinds of things that we were talking about: bacterial pesticides, waste disposal, heavy metal recovery, pollution control, livestock in all forms. Everybody at that point in time was trying to stick a somatotrophin into some animal to change the way that that was going to move. And then that was also sort of the entry for the so-called, the first view of medical farming with the pH, where you were going to have milk basically be the place where proteins were excreted into some animal's milk, recovered in the milk, and then taken into a framework. Started by BST, but then also was an attempted production system. All of that was moving along at this time. I want to take that story of Ice Minus and look at it through three different lenses, because this was really one of the pivotal frameworks. And so this was developed up the road at UC Berkeley. It's a great story, but basically there was a genetic deletion of a single gene that then conveyed frost tolerance on the plants that were inoculated with this Pseudomonas. So the way that that was portrayed, if you can go get one of the images, this was Steve Lindau was the person. Beautiful picture, mountains in the background, all kinds of technology here in the foreground. On the academic view, big picture, just a little bit of words. When the court case finally got the organism into field trials up in the Alameda area, this is how it appeared in Science. You can see this picture of this person in a full Tyvek suit. But you see from the Science perspective, again, a lot of words and a little picture. How the same story then appeared in the New York Times is big picture, little words. Critically, there's something that was to be cutesy. It's a picture of a snowman with the no symbol on it, and the product's called Frostban. And so that picture was trying to be a little illustration of what the technology was. But when you think about how newspapers were printed at that time, zoomed out, this looks scary. And it's got a big no on it. And so now you're trying to tell the public that these recombinant organisms that we're going to go spray everywhere are safe, and that's their first glimpse of it. That suit actually made it into the Smithsonian. It's a fun historical thing. So what was going on in the background and stuff that didn't get there? I want to show this is a scientific advisory panel from 1986. This is for the EPA. This set them up. They had a charge to go figure out how to get data to do risk assessment and safety assessment. This was the charge. So looking at survival, looking at the growth, transfer of material in situ. How does it disperse? What are the environmental effects? What are the health effects? They had charges in 1986 to go look at things that we still have as questions today. And importantly, they proposed that we need publicly owned, professionally managed, contained sites that would be identified for this kind of testing. So early on, commensurate with the coordinated framework, with the guidelines, all of this was going on. Part of it, there's a little link to this down here. When Richard mentioned, these are really hard to find. These documents that are like this are hard to find. What's even harder to find is the work that came out of it. So the EPA had multiple field sites that were involved in different forms of environmental analysis. They were producing proceedings. They were having workshops. They had multiple. These are all various forms of lab-scale containment devices and test systems. And again, to try to find them, you can find various versions. Some are really hard to read. They're just basically black and white scans, and some are in color that are useful. This is one of the dockets. We've moved it up to 1994. This is showing those sites, what was going on at those sites. Again, there were multiple things that were in place. They were producing peer-reviewed data. You can find a few publications on this. There was international coordination going on. Again, to find some of these on the EPA site, you get a black and white scan if you dig really hard. Google's color scans have made some of these possible, but you really have to go and know what you're looking at. This is my final one. This is 1986. This was an internal report that was published, but what I've highlighted here is that they said, in trying to look at assessing risk of GMOs to the ecosystem and health, they said basically, however, there is neither sufficient commercial production nor consumer utilization of these diverse new products to definitively draw conclusions about the ecological effects. What this is important and what this symbolizes, this is about the time that the EPA began divesting the resources. When you didn't have things in the pipeline coming through, they couldn't justify, among other things, keeping those facilities in place. And then just a personal note on this one, the product that we were working on was actually mentioned there. So that's it. I've gone through a lot of stuff, but I wanted to give an overview to kind of go, you know, some things have moved. The systems are not perfect. The systems have lots of challenges. And some things, particularly in the area that we're looking at here, organisms in the environment, they've been contemplated a long time. The questions that we're asking now have been contemplated, and there were efforts to try to answer them from using federal money to try to get those things, but they've all been lost. It's gone. As we try to move forward, even the history of these documents is kind of fading away into 404 errors if you're trying to run through the website. So how we move forward, I think that's what's interesting here. But I'll stop there, and thank you for your attention.